Hi guys, my name is Jessie and today I'm going to be talking about Brilacidin, an investigational new drug. This presentation will cover the innovation and testing surrounding the drug, its chemical makeup, and its mechanism of action. The synthetic structure of Brilacidin has a molecular weight of 936.9 grams per mole, a hydrogen bond donor count of 10, and a hydrogen bond acceptor count of 18. It has 20 total rotatable bonds and a formal charge of 0. The molecular formula for Brilacidin is shown here. Brilacidin is an investigational new drug in the defensive mimetic franchise and has become the leading drug candidate of the franchise due to its current clinical trials, but we'll get into that later. Being a defensive mimetic means Brilacidin is a completely synthetic, non-peptide structure that mimics the effects of natural host defensive proteins. Host defense proteins are the front line of defense in the body's innate immune system. Their biological activities depend on an amphiphilic helix, as shown here, and they are catatonically charged and contain several hydrophobic amino acids. Their biological properties include immune modulating properties and direct antibacterial, antiviral, antifungal, antiparasitic, and anti-cancer activities. This activity is generally directed at critical membrane-associated processes like wall synthesis and cell division, microbial membrane integrity, and intracellular molecules critical to growth and maintenance. Innate immunity is a defense mechanism that involves barriers that keep harmful materials from entering the body. It is the first line of defense in the immune response, and examples include cough reflex, enzymes in tears and skin oils, physical barriers such as skin and chemicals in the blood, and immune cells that attack foreign cells in the body. The innate immunity properties of Brilacidin include antimicrobial effects like antiviral, antifungal, and antiparasitic properties, and also antibacterial characteristics. The direct antimicrobial activity of Brilacidin should have an effect on bacterial, fungal, parasitic, and viral infections. The barrier function includes GI mucosa, respiratory mucosa, and the skin and eyes. The three main goals behind the synthesis of Brilacidin are to maintain a healthy barrier, prevent resistance, and kill pathogens. To maintain healthy barriers, anti-inflammatory and anti-biofilm properties are needed. To prevent resistance, a single dose needs 100% compliance, and the stationary phase activity will kill persistent bacteria. To kill pathogens, a long half-life and post-antibiotic effects are needed. The antimicrobial properties of Brilacidin make the drug a new class of antibiotic and rapidly bactericidal. It has targeted gram-positive activity, a low risk for resistance, and potent anti-biofilm activity. The clinical status of Brilacidin is in phase 2A trials in the U.S. that surround acute bacterial skin and skin structure infections, or ABSSSI for short, with planned 2B studies in discussion. With 14 million outpatient visits for skin infections in the U.S. alone, there is a large market and gateway to serious infections with ABSSSIs. Several targeted indications can arise from them, including respiratory tract infections, bloodstream infections, combination therapy, diabetic foot, and bone and joint infections. The graphs shown here depict the response of Brilacidin to E. coli and S. aureus over 8 hours against a control group of bacteria only. These results support the rapid bactericidal activity that was discussed earlier. An early clinical study of Brilacidin's effect on skin lesions was done, with the result depicted in the following table. A clinical response is generally defined as a percent reduction in lesion size greater than or equal to 20% compared to the baseline, per FDA ABSSSI guidelines. The study compared single-dose and short-course Brilacidin to daptomycin as treatment of acute bacterial skin lesions, and the results support single-dose Brilacidin as the most effective treatment. The key aspects of Brilacidin's mechanism of action is that it is an inhibitor of phosphodiesterase 4, or PDE4 for short. The prototypical PDE4 inhibitor is Rolopram, which molecular formula is shown here. PDE4 inhibitors are drugs used to block the degradative action of phosphodiesterase 4 on cyclic adenosine monophosphate, or CAMP. The PDE4 family of enzymes are most prevalent in immune cells and are predominantly responsible for hydrolyzing CAMP within both immune cells and cells in the central nervous system. As a PDE4 inhibitor, Brilacidin is the first line of defense against infection within the body. PDE4 inhibitors can cause side effects such as nausea and vomiting alongside other gastrointestinal infections, which are most commonly connected and reported side effects. Other big side effects are respiratory infections and urinary tract infections, which have been observed during the clinical use of roflumilast, a medication used to prevent lung attacks in severe COPD patients. Some of the benefits that come with Brilacidin's key mechanism of action being an inhibitor of PDE4 are its potential for use as treatment for widespread respiratory, dermatological, and joint-related illnesses. Two of the respiratory illnesses that are hoped to be eventually treated with this drug are asthma and COPD. Psoriasis and psoriatic arthritis, a common post-secondary illness of psoriasis, are two other illnesses. To recap our mode of action for Brilacidin, PDE4 hydrolyzes cyclic adenosine monophosphate, otherwise known as CAMP, to the inactive form of adenosine monophosphate, or AMP. The inhibition of PDE4 in the mechanism of action results in blocking the hydrolysis of CAMP, which increases the level of CAMP within the body's cells. The company, Innovation Pharmaceuticals, is evaluating Brilacidin in a phase 2 clinical trial as an oral rinse treatment for oral mucosa in patients of head and neck cancer that have undergone chemoradiation. The drug has a high potential for preventative treatment that has been shown in the reduction of severe oral mucosa in those patients. The FDA and Innovation Pharmaceuticals are also testing Brilacidin as an enema administered with water and as a gel for patients with ulcerative proctitis in a phase 2 clinical trial. The findings are supporting Brilacidin as a non-biologic treatment for irritable bowel disorders, with many patients achieving remission. There are also plans in place for an oral tablet as treatment for ulcerative colitis and Crohn's disease. This concludes my presentation on Brilacidin, the innovation and testing surrounding the drug, its chemical makeup, and its unique mechanism of action. Have a wonderful break.